Good afternoon, students. This is Mr. Messina, of course, and I'm here to talk to you about chromosomes. Now, we've been talking about DNA structure, and we've talked about DNA replication as well, but there comes a little bit more when it comes to a macro scale when we look at DNA, and that macro scale is chromosomes. DNA itself is actually divided into separate sections called chromosomes. Now, I'm not saying the DNA the bases and ribosomal sugars and phosphates and all that stuff is broken up into chromosomes, but DNA is a really long molecule, guys and girls. That means in order to keep track of this super long molecule, we have to divide it kind of in our heads into what we would call chromosomes. Now, like I said before, DNA is very long, so we actually have to store it in a very special way. We can't just lay it straight out. It would take too much space, so we supercoil it. Now, by supercoiling, I mean we actually wrap it into around a small ball called a histone. Now, this is kind of a weird thing to think about, so take a look at this. So you're all familiar over here. We have our double helix. This is about what we normally think. So this double helix is spiraling and coiling, and it actually gets wrapped around these proteins right here, which are called histones. Histone. Those are specially designed proteins that allow the DNA to be wrapped safely around it. So if you think about taking like a tennis ball and then take a piece of string and then wrap it around the tennis ball so it's real tight, you're still going to have some extra string, so you have to take another tennis ball and wrap the remaining string over and over again. So you have these histone proteins that take the original double helix DNA and they get wrapped around. A bunch of these histones are also wrapped kind of around each other. They're wrapped into like pins and loops and things like that you can see here. And a bunch of these pins and loops and further coils are wrapped around to make this kind of big spring shape. This is what I mean by supercoiling. This right here. You have tons and tons and tons of DNA wrapped in histones, which are these things right here, which are further wrapped and condensed, and then further wrapped and condensed until you have this kind of spiral that we're talking about over here. A bunch of these different spirals actually are wrapped up until you get this structure looking here, which is what we would traditionally call a chromosome. Now, you see this term centromere here. We're going to get into it a little bit later. The main thing that you have to remember is DNA is divided into chromosomes, and the chromosomes are actually made up of DNA that we supercoil. Now, chromosomes themselves are pretty crazy, actually. Each chromosome duplicates whenever we're getting ready for cell division, also known as mitosis. So, sister chromatids remain attached to the centromere. So that means you start with one chromosome that will replicate into two chromosomes. These two chromosomes are actually attached to each other by a structure called the centromere. So a chromatid is a single strand of a replicated chromosome. A centromere is the region that joins two sister chromatids. So you can see right here, this is one chromosome that's unduplicated. After the duplication process, you now have two chromatids, one chromatid here, one sister chromatid here. Both of these chromatids are the same chromosome. They're joined right here at the centromere, which you can also see there. Bam, centromere. Different organisms can have different numbers of chromosomes. They have a different chromosome number. Now, chromosome number is the total number of chromosomes that you actually have. So you'll notice, like humans, we have 23, flies just have 4, pea plants have 7, elephantes, they have 28. Now, chromosomes are set up in pairs. So humans have 23 pairs of chromosomes, right? So that means we have a total of 46, since it's 23 plus another pair of 23. That, of course, equals 46. Having two sets makes us diploid creatures. If you only have one set, it's haploid. So 23 is our haploid number, while 46 over here, that's our diploid number. diploid meaning 2, haploid meaning 1. You can tell a clean difference here once I actually do a little erasing. So you can tell right here haploid is just N1, which you can see all the separate individual chromosomes here. Diploid is what we actually have, which is N equals 2. So it's a total of 46. Now, here's a good view of what a karyotype is, actually. Karyotype is like a picture of your chromosomes. Can anyone tell the difference between these? I'll give you a second. Which one do you think is the male? Which one is the female? The male one should be on your left right here. The female one, you can tell on the right. You see, males have a Y chromosome, which is right here, where females, they have two X's. They have two X's. And so you can see from these different karyotypes, from these kind of pictures of your chromosomes, you can tell just by looking at them certain things, such as male and female, as you can tell down here. As I said before, a karyotype, it's a photograph of your chromosomes. Homologous chromosomes are paired up. Homologous means similar, if not the same. So for 1 right here, these two chromosomes, they can be very similar, they can be the same, they also can be a little bit different, but we know that they pair at position 1. A lot of these karyotypes here allow us to compare chromosomes from healthy individuals to chromosomes for individuals that are either sick or maybe they have some kind of genetic issue. That's why they're used typically in medicine for diagnostics. Now, having too many or too few chromosomes actually can result in a number of different disorders. Remember that we're diploid, which means we have 2 sets of each chromosome. So we have 2 of each. Now, if you have 1 extra, it's considered a trisomy. If you have 1 less, it's monosomy. A lot of times these kind of disorders can result in miscarriage where the baby is not carried to term. However, these kind of issues can actually cause developmental problems along the way. So a few examples. One would be Klinefelter syndrome, which you can see right here. Here's the X and the Y. You see there's an extra X there. Down syndrome is another one that's pretty famous. If you look at position 21, which is circled right there, there's a third. There's a trisomy at 21. Patau syndrome is a trisomy at 13. You can see that right there. That's number 13. Edwards syndrome is a trisomy at 18. And Turner syndrome is kind of interesting. It's monosomy, which means it only has 1. It only has 1 X when there should be 2. These are just a few examples. There are actually other or different disorders that result from having a different amount of chromosomes. Thanks for tuning in. Be sure to check us out next time. This is Mr. Messina signing off. Have a great afternoon, evening, or good night, depending on whenever you're watching this video.